It's my pleasure to introduce Eric Schadt, who's been to the Davis campus before, and I also want to thank the Storer Family Endowment for supporting these major issues in modern biology series. I've known Eric for now probably about 6 or 7 years, and it's been fun to watch his career grow and expand. As you see in the flyer, he's emerged as a visionary around the world for using large and complex data sets to solve complicated problems dealing with human health and disease. And he's contributed important insights to diseases like diabetes, obesity, and his most recent paper dealing with Alzheimer's disease. His professional career sort of began here at UC Davis. He was a graduate student here for 1 year and then went to UCLA, where he got his PhD in biomathematics. And his first job was director of computing for the mathematical sciences there at UCLA. He's currently a chairman and professor of genetics at the Mount Sinai Medical Center, and also the director of the ICANN Institute for Genomics and Multiscale Biology. In the interim, Eric has served as the executive director of the Rosetta Informatics Merck program for genetics there in Seattle. I met him there a couple of times, or at least one time. I remember telling Eric that I've been following him around and watching him give seminars, and I told him that what I observed was 50% of the people didn't understand what he talked about, 40% of the people understood what he said but didn't like it, and 10% said, "Way to go, Eric." So after 5 or 6 years, that 10% has expanded to 15% maybe? Yeah. So, and after Rosetta Merck, he started an interesting little venture there in Palo Alto called Sage Bionetworks, a nonprofit organization dealing with issues of genomics and health. And then I guess if he didn't have enough to do, he served as chief scientific officer for Pacific Biosciences and helped develop their single molecule real-time DNA sequencing technology there. And as if that wasn't enough, in 2011 in December, I remember at the Smithsonian Institute, he premiered a documentary called The New Biology that he co-produced. And I believe he was wearing exactly those clothes at that premiere. Yeah. So I looked at his CV. It's 190 papers. That's impressive for a short career like yours, Eric. But the most impressive thing is that 35 of those papers were in 2012, and almost 40 of those papers are in Science and Nature alone. So I'm waiting for them to dedicate a volume to you, Eric. So anyway, again, I want to thank the Storer family for providing this venue for Eric to share with us his insights into where genomics is going. And he gives 30 presentations a year, so we're really lucky to have him here today. So anyway, as you can see, Eric's going to be talking about leveraging the digital universe of data to construct predictive models of disease. I'm looking forward to hearing it. Eric, thank you.
Thanks for the invitation to be here, and it's one of the places I get to come now where I don't have to worry about my dress. And you can imagine in New York, although I'm starting to get branded as the crazy guy who wore shorts, and I was telling Ray that I went to give a talk at Goldman Sachs, and love them or hate them, they contribute lots of money. And I go to the top floor, and the head of Goldman Sachs walks right up to me, and he goes, "Man, you must be really effing good to come in here dressed like that." So and I told him, I said, "Well, I guess you're going to find out." So we, one of our big charters at Mount Sinai is how do we leverage this digital universe of information to inform diagnosis and treatment of every patient that walks through the door at Mount Sinai. And of course, we're thinking now more about, not just all the sort of molecular clinical imaging type data, but we're living in a time when lots of different types of data regarding your environments or security or whatever the case may be being collected. And so the idea is, how can we harness all of that information? What are the right ways to harness that information, integrate it with all of the molecular information being generated as well? And how do we end up with these predictive disease models that can help guide decision making in a clinical setting? So very, very translational. Of course, the digital universe of data these days is very, very large. This is from 2011. We were passing 1.8 zettabytes in the digital universe of information. We're now well over, I think, close to 3 zettabytes. And if you don't know what a zettabyte is, right, that's 21 zeros going on there. And that's, think about 60 billion 30 gigabyte iPhones, right? So that's sort of the size, pretty amazing. And so one of our charters is, how do we even get our heads around that scale of information? How do we manage that scale of information? How do we search it? How do we integrate it? And so a lot of the types of things I've been building at Mount Sinai include a pretty high-end data analytics team and supercomputing team. Includes guys like Jeff Hammerbacher, right, who was tapped by Zuckerberg out of Harvard. They were classmates to lead and create and lead the data team at Facebook. And after making all that money and so on at a very young age, he was tired of algorithms for click-through advertising, wanted to do something more meaningful, so has joined our effort. But a number of guys like that who know how to handle this scale of data. And of course, the life sciences is contributing in a big way to this type of information, and this is just showing you the cost of sequencing over the last decade plus. So here's the sequencing cost curve for sequencing a human genome, and here's the Moore's law curve. So this is in a log scale, and you can see the cost of sequencing dropping at a super Moore's law rate. And that's a pretty phenomenal thing to experience in one's lifetime. Not often you see these sorts of trends. If you pull this out, it's interesting to see this curve starting to level off, but I think that's just to, for the third gen technologies to come into full swing, and we'll see that accelerate again. But the bottom line is, this sort of cost drop is enabling us to generate high dimensional data right across large populations for low cost, and again, contributing to the big data revolution. The kinds of sequencing technology I focused on, I won't talk a lot about it today, but I thought worth a couple of highlights, is this third generation sequencing technology where we're looking at single molecules of DNA polymerase in these nanowells and using the DNA polymerase synthesizing DNA as our sequencing instrument. And the advantage that has is speed and long read lengths to help us complete the sort of the puzzle of life. If you think about next gen technologies that have rather short read lengths, if you're trying to put a puzzle together, it can be difficult with just the short read technology. In fact, not only difficult, but misleading. So here's just a graphic where if you're using short reads, the kind of scene that might emerge when you're putting that story together may be very different than reality if you layer in long reads to sort of complete that picture. What you think you were saying may change in a dramatic way. And so our whole aim is, how can we use this newer technology to fill in all of these gaps that exist around the genome? 10 to 15% of the genome is completely unsequenced, right? All the heterochromatin DNA, and as if that's not doing anything important that we need to be worrying about. So the technology is pretty important. We've demonstrated the ability to put these puzzles together using this type of technology. Had a couple of papers in Nature Biotech last year that demonstrated the ability to de novo reconstruct in an automated fashion whole genomes. These are still bacterial genomes, because this technology still doesn't have the throughput of the next gen technology. So it's we're doing what we can, but here showing one of the first examples of a completely automated finishing of a genome, where we literally just generated the data, fed it through the algorithm, and out comes two fully assembled contigs, in this case for the cholera genome that we had focused on from the Haitian cholera outbreak in those 2010. We also recently had a paper, just again to show you how the scene can change if you have the right lens. Here's a paper we had in Nature last year on with Neil Shaw and Kathy Smith at UCSF, where the idea was looking at FLT3 and activating mutations in FLT3 for AML. 30% of individuals with AML carry this activating mutation in FLT3. And so this is a kinase, and so all of the drug companies got pretty excited about that and designed kinase inhibitors to FLT3, and every single one of those failed in clinical trials, and they failed because of lack of efficacy. And so the whole field was about ready to give up on FLT3, started declaring it a passenger lesion, not a causal driver of disease. But Neil Shaw and his colleagues had collected enough data of people on this treatment that when we sequenced with the long reads, what we found were adjacent to the activating mutations post-treatment, that these new mutations that were affecting different codons emerged, and on the background of that activating mutation led to complete resistance to that therapy. So the frequency of these mutations prior to the treatments, where didn't see them, post-treatment, 40 to 70% of the individuals carried these different mutations. But again, that was a picture that you simply couldn't see with short read technology because in a complex mixture, you couldn't put those pieces together. You couldn't link these polymorphisms that emerged post-treatment to the activating mutations. And basically what we showed is we were 100-fold better powered to make that detection by being able to phase that information on single molecules. And so we turned the entire field around that FLT3 is the right target to be pursuing. You just need to design small molecules to not be sensitive to those mutations that emerge. So the DNA sequencing is one thing we've been hitting hard. But deal with all different types of omics data, but also looking at lots of clinical information. One of the big draws for me to Mount Sinai was being in a big medical center with an electronic medical record system that sees 1 million patient visits a year, that I would have access to all of that information. And the kinds of things we want to do is integrate all of that clinical imaging data with all this omics data. And so one of the, I think I have up here, so this biobank we have at Mount Sinai, it's over 25,000, close to 30,000 people now that have been consented for all sorts of testing, both access to their clinical records, sequencing of their DNA, and they don't have to be de-identified. So they've been consented in a way that allows us to recontact them, to link their DNA information with their clinical records, to go into their families, and so on. So it's a very, very valuable resource. And one of the ideas we've been thinking about is how to get the patients like me sort of idea going within a medical center. So what does that mean? So a patient walks through the door at Mount Sinai and you're trying to assess what's going on with this individual. Maybe they have something like heart disease or type 2 diabetes, and you want to understand how best to treat that individual. What you want to do is find the group of patients that all around molecularly look like that patient, and then understand the outcomes of those patients, how they were treated, what they responded to, and so on, and then inform the treatment of the current patient based on that. So what you're looking at here, and this is focused right now on measures, around 50 measures that reflect type 2 diabetes, like risk factors or directly associated with type 2 diabetes. And what we're doing is taking those 50 traits across the entire patient population, the biobank, and we're constructing this similarity map. This is in collaboration with the company IOSD that formed out of Stanford. They use a topological metric approach to forming these sorts of networks. It's a really interesting mathematical advance. If you've not seen it, worth looking at. And so this particular network, each of the nodes are individuals in the biobank at Mount Sinai. And again, we're connecting them with edges and grouping them in this topological space based on their similarity across these 50 different dimensions of data. And we've now color-coded this network according to diabetes status. So here, if you're red, you're at high risk or you have diabetes, versus blue is you're far away. And the first thing we see are these different clusters emerging in certain parts of the network that are reflecting diabetes. But now, since we've exome typed all 25,000 plus individuals, now we can go in and say, well, what are the variations that may explain differences in these groups? And this is all happening real time, right? This isn't a formal GWAS study where you're going through all the rigmarole of that kind of thing. This is, you're just looking at these things, identifying the different groups and saying, give to me the variants that explain differences. In this case, we get this top hitting variant for this calcium channel gene with a pretty good p-value for association that discriminates, so discriminating between this group A and group B. What's interesting about this particular gene is it has been implicated for type 2 diabetes in several different studies, although the support we're providing at this point is stronger, basically given the bigger numbers we have within the medical center. And what's interesting is this is related to calcium flux in a given system. And as you might know, people with type 2 diabetes tend to have lower calcium levels, and a lot of the comorbidities of diabetes are thought to stem from that lack of flux in calcium. And so when we now color code, we say many of these patients have had blood panels run, right? We have access to the EMR, so we can go into the EMR and say, give me the calcium measures for these patients and the blood panels they had run for whatever reason. And now we've color coded by calcium levels. And here what we're seeing is this group here having very high calcium levels, and this group over here having very low. And so now what you're immediately seeing is a stratification of that patient population into different groups that may have increased susceptibility or severity or risk of comorbidities based on lower calcium levels. And now we can go in and say, how were those patients treated? How were these patients treated? And were the outcomes different depending on how they were treated, and so on, and ultimately want to feed that information into the hands of physicians. So that's just sort of an example of how this real time, how handling this big data in real time can be, have clinical impact, and that's kind of what we're moving towards. But what we want to do is not just look at that clinical information, right? We want to go into the big universe of all the different omics information and ultimately build these sorts of predictive models that reflect all the different molecular information, clinical information, imaging information, whatever information exists at population scale. We want to build these models that reflect causal relationships that get us basically towards understanding what's going on in a given patient. And the one important thing I wanted to highlight was how important going beyond the association space is. Like, we want to get at causality. That's like a theme I've been beating on for a really long time, but it's worth continued beating because it, I think, while it has some traction, not fully appreciated by all. And so the way this works, the way we handle this in this population-based setting is we're looking at lots of different traits, right? We're looking at traits, sampling them in the population. And the first thing you'll notice as you're sampling through the population is you'll notice that these traits are correlated, right? So that correlation becomes interesting to you. And we've seen the correlations have broad clinical impact, right? If things are correlated with an outcome, you can maybe use that as a biomarker and predict response or look at prognosis and so on. But this association alone doesn't get you to causality. And what you do in your mind is you form a hypothesis as a biologist, and you say, well, I think this correlation relationship is causal in this way, and then how you're going to test that is you set up this experimental system where you set up the system, you artificially perturb the gene that you're hypothesizing is causal, you perturb it, you see the response, and then you look for a subsequent response in the other gene or the other trait. And based on that sequence of events, you'll declare a causal relationship, right? So you're knocking the gene out, you're overexpressing it, you're chemically perturbing it, whatever your method is. This has been the mainstay of biology for a really long time. But what we've done is to leverage DNA variations that occur in the population as our source of perturbation to infer causality. So we're leveraging DNA variation as the perturbation source to knock down or overexpress your molecular traits, and based on that, see what causal impact that has on higher order traits like disease. So this is the idea of what's now referred to as Mendelian randomization. And I'd sort of first published on this in 2003 in Nature, had a network-based version of this in 2004. And the way to think about how we can get causality out of the DNA variation as a perturbation source is think of a clinical trial. So this is just like setting up a clinical trial, where how do you establish causality in a clinical trial? Well, you have treatment arms, you have like a placebo arm and a treatment arm, and you randomize patients to those arms and then on the treatment, and then differences you see between those, you can infer a causal relationship between that drug, the action of that drug, and the endpoint of the trial. And the thing that allows you to do that is the randomization of those patients to the treatment arm. Well, we can play the same game with the DNA, but now instead of randomizing to a treatment arm, you're being randomized to a genotype arm, right, through Mendelian segregation of those chromosomes. So basically, we can take the population at any given locus of DNA, and we can stratify the patient population into different genotype groups. And then we know you're randomized to those genotype groups through Mendelian segregation of the chromosomes, right? So the nature has handled the randomization mechanism for us. So we basically say you've been randomized to these different treatment groups, and now we look for the treatment groups that are correlated with the traits of interest. And based on that pattern of correlation, there's only one of three ways that the two traits can be related. So remember, we're starting with DNA variation as a perturbation correlating with two traits of interest. And given that kind of setup, given you've been randomized to the treatment or genotype group, there's only one of three ways these two variables can be related. So either changes in DNA cause changes in phenotype 1 and then phenotype 2, or vice versa, or the variation independently affects both traits. So we can mathematically model that and then simply pick the model that best supported by the data and make our causal inference. But again, we want to do this beyond single pairs or pairs of genes or pairs of traits. We want to go into the entire space. So we use what's known as Bayesian network reconstruction theory to systematically go through the entire space of DNA variations and molecular traits and clinical traits. And the aim is to form this sort of model, right, where we have DNA variation, we have all our different molecular traits, we could have clinical traits in there as well, and we're basically systematically going through and understanding all the different causal links using DNA variation as a perturbation source, and that constructs local networks, but we of course look over many different tissues and systems, and so we see this network of networks, and it's really this network of networks that's taking us to a fuller understanding of these biological systems, right? So and then we want to use these models as predictive simulators of biology, right? So this now reflects a lot of biology, so we can understand how changes in one part of the network cause changes in another part of the network, and this may be in a given cell or a tissue type. But again, we're modeling over the entire system, so we want to look at how this information is being communicated across different organ systems. So again, the modeling enables us to do that. And the modeling, again, because it's predictive, we can do simulations, we can knock genes down, we can overexpress them, we can see how that information propagates through the system, and ultimately understand how to better target and diagnose disease. So again, the ultimate aim is predictive network model, and the predictive network model is going to help us identify what are the key nodes that we want to be targeting or tracking for therapeutic intervention, or for assessing risk or progression or severity. All of those things become possible with this type of modeling. So the DNA variation is one source of perturbation that we can use to make these causal inferences, but time series-based information is another way that we can make this type of causal inference. And so this is just to highlight that in addition to the DNA variation information, we also use time series-based information with dynamic Bayesian networks to put those networks together with that type of information. So before I launch into a couple of examples, I'll just show, tell my sort of flow of how this is really going to take us beyond the biology of yesterday, right, where we focused on single genes, on single products of those genes, single coding products affecting single phenotypes. So this was the very reductionist view where we wanted to believe that simple linearly ordered pathways were how we could explain complex biological systems. Today we're in this omics game where we're looking at whole genomes and transcriptomes, proteomes, epigenomes, and so on. But we ultimately need to organize that information in ways that we can understand and predict. And so in order to do that, we need to get at this causal molecular network type inference to actually progress biology beyond the more naive view. So to give you some examples on how we've done that with some of this big data, I'll start with this paper we had in Nature, or I was on with many others on the IBD consortium. And they had initially, basically came to me because they had identified in this consortium 163 loci for IBD that had, so genome-wide significance. So these are 163 causal, variants that are, leading to IBD or predisposing to IBD. And the question was, well, what do we do with that data? We don't know what all the genes are. We don't know if the variant's activating the gene, knocking down the gene. We don't know what pathways these things necessarily operate in, and so on. So what do we do with this information? So what I did was I took these 163 loci, and then you want to project them. So we build, like the type of networks I was showing, we basically construct these networks off all the data that exists in this digital universe. We build these models, and so this is one such model. This model was constructed from bariatric bypass, 800 and some odd patients who underwent bariatric surgery. This is the omental fat network from those patients, so 800 plus patients, all had the RNA profiled in omental fat, sub-Q fat, liver, and stomach. All had the genotyping done genome-wide at high density. And then we build this network. So all the blue nodes and pink nodes in this network represent either gene expression traits, DNA variants that affect those gene expression traits, clinical parameters, and there are a couple of other molecular things like cytokine panels and so on. In the green are the different edges that reflect relationships between these traits. But remember, those edges are all directed, right? So they're all reflecting causal information, hard to see here. But what we do is we take these networks and then we project onto the networks these 163 loci. So I have literally hundreds and hundreds of these networks. We project across all the networks. And this network stood out because those 163 genes were isolated to, or a big majority of them were isolated to this very specific part of the network. So the pink nodes are genes that are in that 163 IBD study falling in this very specific part of the network. And the chance that you see this sort of pattern of co-localization of those hits to one really specific area of the network is very, very small, less than almost zero, effectively. And so now now what do we do with that? This looks like, again, the big hairball, and what do you do with that? But remember, this is a causal probabilistic structure, so we can carry out simulations on this data. We can knock each gene in that network down, we can overexpress it, we can assess what the effect of the network is, we can do that in combination. And since that's done in silico, we can do it in seconds on supercomputers. So that's what we do. And now I'll zoom in to one of the views we have of this network. This is a zoomed in view of those pink nodes that, remember, were very significantly enriched for IBD genes from the GWAS study. And now what you're looking at is these nodes right here are the causal master regulator nodes for that subnetwork that we identified through simulation by knocking down and overexpressing all of the genes. And then we're looking at the genes that have the biggest impact on the state of that subnetwork, right? And we call those our regulators of the network. So those are these big nodes. Then this first link of genes to the key regulator nodes, these are genes that are within a path length of 1 of the key regulator nodes, so they're most proximally affected regulation-wise by this layer. And then these genes out here are greater than a path length of 1 away from the causal regulators, so they're more downstream. And these guys here are sort of interesting because they are causal for the regulators, so they're modulating the regulators. And the interesting thing about being able to dissect the structure of this network is now you can start understanding how you might track for diagnostic purposes, what are the right nodes to track for assessing risk and severity, and what are the right nodes to target for therapeutic intervention. And just to tell you how this structure matters, that network itself was around 10 to 15-fold enriched for genes that harbored variation in the human population that caused IBD. But if you look at these genes are around 30 to 40-fold enriched for harboring variants that lead to IBD in the population. These guys are around 10-fold enriched, and these guys are around 5-fold enriched. So these guys clearly are having a much bigger impact, from variations that occur in the population to causing IBD. And then we can look at that information in another way to really understand how is the network communicating with itself and with others. So this is a 3D Circos plot that now what we've done is we've organized the nodes by biological process. So these different triangle colored regions reflect different biological processes, and we've organized the nodes into these different colored triangles depending on whether they've been annotated for that process. But now what we've done is we collapse edges between nodes into these arteries if they have common paths through the network to other nodes. And what you see emerge when you do that are these big arteries, and they start telling you how different biological processes, what they're communicating with, and what their impact is on other biological processes, so we can start sort of understanding the information flow. And now we want to take all of this and actually get to something that matters, right, for the patients, maybe a new treatment, right? So what we do there, one of the guys I convinced to come from the sunny West Coast to New York City is Joel Dudley. He was out of Stanford. And he had pioneered this approach where we can take the disease network that I was just showing you, and we can intersect it with the compound treatment signatures that get generated, say, like in Connectivity Map or Cell Encyclopedia, right? These different resources that have given a drug to different cell systems, looked at the transcriptional response. We can take that information and we can intersect it with the disease networks to basically see what are the compounds that push the key drivers or different players in that disease network in the right direction to transform that network into a more healthy state, right? So we can run, again, this is all in silico, right? So far we've done no experiments. We've built the network, we've made the predictions, we're now linking it to a new drug, and now the proof is in the pudding is in the eating. So Joel had identified topiramate as one of the top drugs that's hitting the IBD disease-associated networks. And so topiramate, right, this is an anti-seizure drug that how many in the room had thought this could help IBD, right? Nobody. And so here's a drug that had no indication for IBD, for these inflammatory processes, yet this purely in silico-based method said that this would be the case. So what happens when you drop that drug into mice where IBD's been induced? So here you can see this chemical perturbation in the mouse that induces IBD. And when those mice are treated with topiramate versus the market-leading steroid for addressing IBD, you actually see as good or better a response from the topiramate versus the market-leading compound for IBD. Right, so again, a completely novel indication on a drug that already exists that now can go into clinical testing in these individuals. The other thing that became sort of interesting to me about this particular network that I've been talking about for IBD is its association to many different diseases. And so we recently were focused on Alzheimer's disease, had a paper in Cell a week or two ago where we were basically looking at many different tissues from Alzheimer's and non-demented control subjects, looking at prefrontal cortex and cerebellum and visual cortex. I think we had around 800 individuals. We had their different pathology-based data on these individuals. We RNA profiled all those different tissues. We did genome-wide genotyping across all those individuals. To basically go through the same flow I've been talking about to build these association-based networks, integrate the genetic information to get at causality, and ultimately build these predictive models. The one thing that was sort of interesting about the Alzheimer's was this idea of differential connectivity that came into full view. So here what you're looking at is the co-expression network across that population. And so these different, this is a topological overlap map view, and these different colored bars indicate different highly interconnected subsets, what we call modules or subnetworks within that brain prefrontal cortex network. And if I zoom in to the different colors here, what I'm plotting here is the subnetworks. And basically your red, so it's basically genes against genes, and you're just looking at the interconnectivity of those genes. So think of it as correlation of correlations. And you're red if you're highly interconnected, so very tightly connected, and you're white if you're not connected at all. And what I've done, since this matrix is symmetric, on the upper triangular, I'm plotting the Alzheimer's cases, their connectivity pattern in the brain, and on the lower half, I'm plotting the normal. And what was interesting when we looked at the difference in just gene expression between prefrontal cortex and Alzheimer's versus prefrontal cortex and non-demented controls, we were expecting, right, that's the area of the brain that's being torn apart, and we just expected to see massive expression changes. And we actually saw like a few hundred genes. It was like one of the smallest signatures we'd seen in a big disease study like this. So that was disappointing. But when we looked at this differential connectivity, so here you see just striking patterns of differences in the connectivity of these genes. So even though their expression is not differential between the groups, how they're connected, how they're correlating with one another in the network varies dramatically. So while there was only a few hundred differentially expressed genes, there were many thousands of genes that were differentially connected between the Alzheimer's and controls. So again, that idea of how the network's wired maybe being a dominant feature in this particular disease. And one of the networks, so again, we screen these different networks against other existing networks to look for associations to disease. And the network we found here that was highlighted in the paper was nearly identical to the IBD network that we found and that was in the Nature paper. So this was sort of interesting that if, the same network, so this deals with immune function and inflammation, and so no longer really a surprise that this is a common feature in these diseases. But in this particular network, something like 40% of the genes that have been identified in the GWAS for Alzheimer's fell in the single subnetwork, a massive enrichment. And some of them were recently identified. TREM2 was a gene that Decode had identified, was a paper in New England Journal, and I'm forgetting the other group. They had back-to-back papers, but rare variants in TREM2 cause Alzheimer's. These are sort of emerging from the GWAS and whole exome sequencing studies. So again, a network that's enriched for variants being found in the GWAS, but what we're providing is context, the actual molecular mechanisms that are underlying that disease. And remember, this is causal. This isn't association. This is showing a causal link between variations in this network in the disease. We had identified TYRO-BP as the key master regulator of that network in this particular disease state. And it was interesting that, again, this big finding from Decode and the other group, TREM2 actually signals through TYRO-BP. So again, that was, came out while this paper was in review. So it was a good confirmation that we had identified the right network. But again, we provide the whole molecular setup of what's going on in this disease, potentially. And then because this, so now we're seeing this network connected to IBD, we're seeing this network connected to Alzheimer's. I carried out another experiment with colleagues at Merck where we basically generated expression signatures across a whole range of diseases. So we had disease models in mouse, and then we were comparing the relevant disease tissue in the disease model versus the control healthy model. And so we did this for atherosclerosis, for diabetes, for inflammatory pain, COPD, asthma, fibrosis, and so on. So like 11 diseases, generated this signature across all the relevant disease. So the tissues are different, but it's the disease relevant tissue. And then we basically looked at, so what you're looking at here is the heat map plot of the genes that are in the network that are associated with IBD and Alzheimer's, and you're looking at the expression of those genes over all these different diseases. And what you see is a pretty amazing pattern of concordance of the state of this network across these different disease conditions. So again, implicating very strongly this particular network is being maybe a key connector across all these different disease states that we know just from clinical features are connected in patients. So then what we do, this is sort of getting to the to-peer-mate game. Now we want to understand how do we actually target this network to have a better clinical impact. So what we're starting to do is look at these sorts of maps where this is the special network I was just talking about that's implicated in all those different diseases. So the blue nodes reflect that network. And now what I've done is I've taken all marketed compounds, all compounds that are on the shelves in pharma that we could get the chemical structures for in some natural products, and worked with a C-Change, this group out of UCSF, that basically has mastered how do you predict these small molecule interactions with proteins, the products of coding products of these genes. So what you're seeing here, the green nodes are all those natural products, marketed drugs, and so on that are known or predicted to interact with the different genes in this network. And the pink nodes below are the diseases that have been associated with high confidence to different nodes in this particular network. So again, this is starting to give us a way more holistic view of how we need to be viewing disease biology and how best to treat it, because what you can see are that there are many, many drugs that could be impacting the state of this network. And of course, depending on how you hit that network, right, if you're disrupted in the brain, you're getting Alzheimer's. If you're disrupted in the gut, you're getting IBD. If you're disrupted in the joints, you're getting RA, and so on. Like so how you target this and in what condition is going to matter. And so we've started playing around with these kinds of maps to try to lock on to better combination therapies to do these treatments. And what I'll show as a final set is, the work we're doing of the same ilk, but now in cancer, to basically get at a personalized cancer therapy for patients based on the type of cancer that they have. So this, again, is going to play the same kind of game, but this is now much closer to real-time decision-making and impact in patients that have cancer. And we're basically taking cases where they're terminal, they're not responding to frontline therapies, standard of care, or they have forms of cancer that are untreatable. And what we're doing is we're taking tumor RNA and DNA and sequencing those whole exome and low coverage whole genome, and then sequencing germline. And then we're taking that information, and unlike a Foundation Medicine or TGen or MD Anderson, where what they're doing is they do the sequencing, but they're just projecting that onto known variants that are known activators of PI3 kinase or PTEN or KRAS or whatever the known signaling genes are. So they have maybe a list of 200 things that they look at, and if they see a match, then they'll make the call, but 90+% of the time they don't make any call because it's not one of those genes that's mutated. So what we do is we take all that information and we project it onto the types of models that I just showed that we've built up across different types of cancer that's in the digital universe. We integrate it all, build these predictive models. And once we have that projection, we identify what subnetworks are disrupted in those patients, in a given patient, what are the key drivers. And then what we do is we have 3 different treatment arms that we could personalize for that individual. The first arm is we do a chemigenomic screen against the network that's both functional and structure-based. So we do a structure-based chemical screen where we're saying what are the known drugs or any drug that's existing in any trial that's known to target any one of the genes in this network or predicted through this off-target effect modeling from sea change. But we can also do the type of thing we did for topiramate, where it's a functional screen. It's looking at the transcriptional readouts of these compounds and intersecting that with the networks. And based on that, we could come up with a novel treatment strategy. For some of the models, like head and neck cancer, so we're basically doing head and neck, multiple myeloma, ovarian, and breast. For head and neck, in some cases, we can actually take the key drivers that are disrupted, multifactorial disruptions that are existing in a patient, and we can make a fly model of that tumor, of that patient's tumor, and then do high throughput screening on that fly model and take hits from the fly model and take it into xenograft models from that patient. So it's a way, the flies are very fast. We can do that in 2 to 3 months, but basically survey the entire space of drugs and combinations of drugs that are, can hit multiple components of the network to come up with a novel therapy. And then the third is we do CD8 epitope predictions on the genes that are disrupted in these networks in the tumor, specific to the tumor. So identifying immunogenic genes specific to the tumor that we then design with Carolina Polovic at Baylor Health, design novel vaccines that can be turned around in 8 to 12 weeks. So each of these are sort of novel, completely specific therapies guided based on what's happening in that patient's network. And just to show you one of the cases, here's a glioblastoma case. So I mentioned the big areas we're covering, but we'll take sort of interesting cases, and one was a glioblastoma case. Glioblastoma was interesting because there's lots of data in the digital universe to build good models. So again, we take that patient's, generate all the omics data, project it onto the model, and then what we're doing is looking at networks that are enriched for those mutations. So the glioblastoma network, there are 552 samples in TCGA of glioblastoma, and 150 or so of those have been sequenced. All have been RNA profiled, and there are CNVs for all of those. So we basically constructed the network as about 10,000 nodes in the network. And when we projected the patient's information onto there, we found a network of about 1,000 genes, so 10% of the network, that were super enriched for variants that this individual harbored. In fact, greater than 50% of all the mutations we were seeing in this individual, high impact mutations in this patient's tumor, were localized to the single network. And the probability you see that by chance, of course, very, very small. So all these blue nodes are ones that are mutated. And now we do the chemical genomic screen, a functional screen, against that network, against all existing compounds that have molecular readout information. And then here's just showing you how we would play the game of getting to the therapy we would recommend for this individual, and how the structure of the network really matters when you're doing this. So here's 3 drugs that are used in cancer therapies that were part of our screening. And what we're doing is we're looking at the molecular readout of these drugs against that network. And in the first case, we're rank ordering. So here you're looking at the ranking of these drugs, the many hundreds that we test. The ranking, basically, you're at a high ranking if you're hitting the network really well. And so what you see here is if we look at the cancer network model in general, you see that all of these drugs are sort of falling in the noise of the rank ordered list, so we wouldn't pay attention to any of them. If you look at the patient-specific subnetwork that's disrupted, you see this trend of increasing ranks across these drugs. And if you look at the key drivers, again, remember the key driver analysis we can do on these causal models, what we find is this irinotecan jumps all the way to the very top. So if you looked at the entire network altogether without any patient information, it's in the noise. If you look at the individual specific changes in the key drivers for that individual patient, this is the number one drug. And so now and what's interesting about irinotecan is it was thought to be potentially advantageous for glioblastoma, but those trials failed. But again, what we're doing now is saying for this particular patient, this particular drug may work. And so this is just an example, and I unfortunately do not have for you the punchline of what happened in this patient. That study's still ongoing, so a lot of this stuff is pretty new. So, but that's the kind of game that we're playing, right? And just so you get a handle on the speed, we are basically doing generation of all that data. We're doing rapid runs on the HiSeq 2500, so from receiving sample to getting FASTQ files off the sequencer is about 36 hours. And then it takes us around 3 days to carry out all the analysis. So literally in about a week, we have what we would recommend course of treatment for a given patient. And so just wanted to close by saying that we're doing this across many different diseases. Cancer, there's lots of existing data, but we're doing lots of studies where we're collecting very high dimensional data across a large number of patients. And our ultimate vision is one of connecting all of this information and providing very detailed information to the individual of how they would navigate their health course, given the makeup of their molecular self, in addition to environmental components in their daily lives. And so the type of modeling we've done, this is the final slide showing you how we vision, this is down the road, so this is a real model. I'll tell you how it works, but it's not as general as we need to have it on your smartphone to help guide your health course. But we ultimately view this type of topographical map and the different trajectories you can be on this topographical map is being displayed to you on a daily basis, where we can tell you which trajectory you're on, and if it's a good one, how to stay on it, if it's a bad one, how to get off it, whether it's changing your diet or exercise or taking small molecules or whatever the case may be. So what this is showing is basically we've taken the different networks we construct, say for the Alzheimer's, for the normal and the disease case. We project those into this 2D space where every XY coordinate now reflects a state of the network, of the different networks that we projected. And the z-axis corresponds to the probability of being in that state given the makeup of the features that are reflected in the network for a given individual. And so what you see are these high probability peaks for these different states reflecting normal conditions and disease conditions. And since, again, these models are probabilistic causal models where we can play the game of in silico biology, the different trajectories that exist between the states define the constellation of genes that you would target, and not only that you would target, but how you would target them in a quantitative fashion to move you from one state to the other. So the trajectories are literally defining, like here are the 4 genes that you need to target. For this gene, you need to knock it down 10-fold. This other gene, you need to boost it up 5-fold. Like it will give you that sort of a prediction. And the hope is that once you identify which trajectory you're on, whether you want to stay on that trajectory or jump off, we would be giving you advice on what you need to do to jump out of a given state. So again, I think that's definitely not there today, but for the future, that's exactly where we see this going. Again, how do we leverage all of that amazing information to provide you real-time information on how you might live your day-to-day life or how to stay healthy. So with that, I just want to thank the rapidly growing group at Mount Sinai. I've hired like 100 people since I've been there, so this is some of just the key PIs. I'm still very much interacting with PacBio in different academic labs in the New York area. We have good relationships with the University of Bonn, carried out if you read the Cell paper, the TyroBP validation experiments. They were awesome in nailing that all down. And Sage BioNetwork still heavily involved with that. So thank you for your attention. Sure.
Brilliant, Eric. You've come a long way from Appy Chips and Roche, that's for sure. Remember those days?
I do remember.
I would suggest actually in your last heat map you could have another peak focusing on optimized super health to go that way. But actually my question is the integration of networks not necessarily are all equal even at the molecular level. So you're doing really well with the express genes improvements, but there's really interesting stuff in the non-coding control regions, the non-translated RNA, the microbiome.
Right.
I mean, all of those components, just to how to integrate those in a way that will make sense for therapeutic targets, personalized therapeutic targets, I'd imagine.
So it's a great point. And first of all, I'll say that the method of doing the constructions and integration is agnostic to what kind of data. So it's it's like pour it on. If you can, several of these nodes are non-coding RNAs. If you're doing metabolite profiling, we publish papers on proteomics, metabolomics, all of that being integrated. So the model can take it up. It's just a matter of is it cost effective to generate it. And why we think it might not be necessary is, again, these networks, again, what you're seeing isn't exactly right? It's not as if that network's 100% predictive. It's just you got enough of the pattern to recognize what's being disrupted and hopefully enough of the key drivers to say, here are the key drivers that are actually modulating the state of that network, and now your screen, again, is just looking for patterns. So the beauty of it is all pattern-based. It's not dependent on having every single node exactly right. It's just, do you have enough of the pattern to make the lock? And then, yeah, absolutely. So one of my first, or not one of my first two hires, but one of my best two hires, to Mount Sinai was Jose Clemente. So he was, Rob Knight's star postdoc, first author on all Rob Knight's amazing papers, and Jeremiah Faith, who was Jeff Gordon's star postdoc. So we are hitting the microbiome very, very hard. In IBD in particular, we'll have an announcement in a week or two on that. And one of the things we've done in the microbiome with Harm von Bockel and Jose Clemente is we've worked out the assay to do RNA profiling of the microbiome and integrate all of that information into these networks. And again, that's just a big data game. And the human subjects, we're getting like 500,000 to 1 million transcripts from a single microbiome sequencing, RNA sequencing run. So again, how to handle all of that information and how's it interacting with the host, and what we can learn is like a major area of study for us right now. Yeah, in the back.
Yeah, it's fascinating. Now, some people consider like the Erlinger paper in the Nobel journal, where they were looking at the cancer component, different sites of the tumor, even primary versus metastasis, tremendous heterogeneity.
Yep.
How do you take that into consideration when you're doing a sample size where you're normalizing based on that one sample area, and yet at the same point they were showing actually within one patient, lack of crossover in some of the mutations. So how do you take that kind of complexity?
Yeah, that's a great point. So the extent to which the minor forms of that heterogeneity are driving things, we're at risk of missing. And the way I look at this more is we're setting up a system that can scale to, like one of my other big projects is IVF-related pre-implantation diagnostics, which is really helping us drive the technology of single-cell-based assays. So we're popping a few of the cells out of the embryo and doing RNA-seq and so on. And so really preparing for the day where the single-cell-based assays are able to run in a cost-effective way. Today, we simply can't afford to do multiple biopsies from the tumor and sequence everything and do it. It's just not cost-effective. So what we're hoping to demonstrate here in the clinical trials. So what are the clinical trials we're running? Well, so in multiple myeloma, the doctors have, and this is enthusiastic oncologists who come to us, and they're saying, nothing I do for this class of patients is going to matter. They're going to be dead in 4 to 6 months no matter what I do. So sure, if you tell me based on this what to give them, I will give it to them. And so we're randomizing patients to the arm that's informed this way, the arm that's standard of care, and we'll see whether there's a difference. So we're just trying to get at that if we use this sort of decision making, even though you're right, it's grossly incomplete, it's going to improve outcomes. And then as the data generation gets better and better, more cost effective to do single cell, then then we can play the kind of game that we need to play to get at your question.
Well, you're right. Also, the FLT3 data was fascinating in the sense that if you can pre-select or use the model as a predictor for what could be mutated downstream, that's where it's going to go.
Yeah. So isn't this kind of classic issue, though, that even though you could know a lot of things about the disease, that doesn't always help you treat it? So it seems like at the end point of this, to try to improve health outcomes, you're still limited by, let's say, available FDA-approved drugs.
Yep.
I mean, of course, you could do a high-throughput screen in flies, but I don't know how you could get a hit from that into a person on a time scale that could really help them. So if you're limited to the FDA-approved drugs, for treatment, I mean, isn't that a massive limitation to whatever?
Yeah, so absolutely. So these are all great points. That is, this isn't yet the holy grail, but it's, again, the point is, does this take us further than we would be anyway? Why did we choose head and neck cancer? It's because it's a very slowly progressing cancer. So it's terminal. The patient, if you got head and neck, you're going to die of head and neck cancer, but it takes 10, 15 years. And so in that case, we can turn around, right? The fly model takes us 2 to 3 months, the vaccine's 2 to 3 months, the xenograft model's 3 months or so. So that in the course of a half a year, we can get to a very personalized therapy for that individual. And again, we're doing that trial to see whether, and again, the marketed drugs, it's not just the marketed drugs, there are kinase libraries, that have been through safety testing, and so there are lots of assets that are residing within companies like Roche and Merck, and who's your guy close to here, Patrick Sunxiang, who made $7 billion off of Braxane, who now bought back the kinase library from Celgene and wants to play this kind of a game. So there, in addition to the marketed drugs, there are maybe 1,000 to 2,000 other drugs. And the question is there a combination that you can give those in that are going to systematically hit the right kinds of pathways simultaneously to wipe out the cancer so it doesn't recur? So I like the combinatorial nature, which is going to take us further than what they can do today, which is just making up what they might want to give in combination. But you're right, ultimately it's got to be, can we customize, are we going to get to the point? That's why I like the vaccine part. We can design a novel vaccine, like the mutation that occurs here, that's immunogenic, like that may be the only person on the planet that carries that mutation, but we can design that vaccine specifically for that, to target that particular epitope and see whether it benefits the patient. So I think, again, the question is, if we're using all this information, can we enhance outcomes? Because I think if we can show that, then the idea that, hey, we should be leveraging this big data in a more routine fashion will take hold and just become a part of what medicine becomes. Yep.
Follow on to that, what about diet, things that you could eat more of that might help? I would imagine there's a lot of different foods and things that might help in this process.
Yeah, we've been one of the connectivity map like resources we're generating right now is based on natural products. So, love that area. So we want to see, what are the different compounds that occur naturally in different food types, and what molecularly are they doing, and then map those onto those networks and play the same thing as a drug, but see whether we can do that. And we do a lot of diet type studies where, again, that network I showed you that's connected to all those diseases, the thing is profoundly affected by diet. So again, those trajectories that are showing it pinpoints genes that you could perturb, but you can also look at environmental stresses and project those onto. So you're exactly right. I think it's ultimately going to be a way more holistic set of recommendations. That may be diet changes, it may be exercise, it may still be some drugs, but there may be natural products that are, you can just enhance in your food or whatever. Yeah, we recently formed a collaboration with Berg Pharmaceuticals. Most of you probably not heard of Berg. They're out in Boston. But Carl Berg, he's California dude, very rich guy who backed this thing. And they've set up a pretty high throughput assay center for doing proteomic, metabolomic, glycomic, and lipidomic profiling. And we've started working with them to try to generate that sort of information in a more consistent, high throughput way. So the problem with the proteomics has just been technology. Like it's just not at where the DNA and RNA sequencing is at. So, but again, the methods are agnostic. Doesn't matter.
Looking at the human diseases the way you're looking at them, how has that sort of informed your look back at the animal models to sort of decide whether or not you're actually modeling what you think you're modeling?
Yeah, it's a good question, and I guess what I see in my own work is this evolution towards all human. I mean, we still look at a lot of these networks are based on animal models, but I find more and more the kind of data we used to think we could only get in mouse, we're now able to get in human. But it's still, so it's, we still, I definitely am not going to give up on the experimental systems. I mean, you saw the fly stuff, we're doing the xenograph stuff. And basically in those populations, because you can control so many other variables, like the reliability of the networks are better. So the question just becomes, how well do those recapitulate the mechanisms going on in the human? And so we have a lot of work going on to assess that. And so when we can use the mouse network, we use it just because it's more reliable. All right.
Okay, so many patients come with partners. Partners would have similar environments, similar lifestyle choices. Looking at the partners could take out quite a bit of the genomic noise. Do you consider that?
Yeah, so have not considered that. And for the cancer cases, I'm not sure because the tumor genome is pretty important. But for the, so great. So for the one study I have here that I wanted to talk about, but so chronic fatigue syndrome, we're doing 250 cases, 250 controls where many of those are matched, like the partner, because they do share this environment. In chronic fatigue syndrome, right, we don't really know what's going on. It's a pretty horrible thing if you've got it. And then we're isolating T cells, B cells, monocytes, dendritic cells, NK cells, RNA seeking those all pre and post exercise stress, gut microbiome, and so on. And have basically pulled those matched, unaffected controls in from the families exactly for that reason. So I think it is a great idea. And it's it complicates a study to do that, but yeah.
They come at the same time in the clinic, they can
Yeah.
use the same kind of consent forms. In terms of
Yeah.
practical issues, it
Yeah.
might be feasible.
Yeah.
I know you guys have a lot of questions. We'll be hanging around here for a while. But anyway, I want to thank you, Gary.
Okay. Yeah. Thank you.